It's my pleasure to introduce Dr. William Fraser Symmans. He is a professor in the departments of pathology and translational molecular pathology at MD Anderson Cancer Center, where he practices breast surgical pathology and cytopathology. He also leads a research program in breast cancer pharmacogenomics. He received his medical degree from the University of Auckland in New Zealand, where he also completed, and then he also completed his residency at Columbia University in New York. Did also his fellowship at MD Anderson Cancer Center in Houston. Dr. Simmons has joined the faculty of NYU Medical Center in 1993 and then later came here to MD Anderson in the year 2000. Dr. Simmons' research focuses on breast cancer with specific emphasis on neoadjuvant treatment trials for evaluation of chemosensitivity and endocrine sensitivity, assessment of response to treatment and development of diagnostic tests to select the most effective treatments for individuals with breast cancer. His other major research focus is addressing the effects of biopsy sample quality on genomic test results in order to establish appropriate best practices for clinical diagnostic use, particularly with small clinical biopsies. His other responsibilities include being the director of Translational Research Program and the executive committee member for the Alliance for Clinical Trials and the Alliance Foundation Trials, and is also a steering committee member for the Translational Breast Cancer Research Consortium. The title of his talk today is Development of a Clinical Assay for Breast Cancer Using the Invitrogen QuantiGene Plex Technology. Dr. Simmons?
Thank you. Thanks, Bobby. It is very interesting to be giving an in-person talk. It's been a very long time for all of us, so it's wonderful to be together collectively. My work, as Bobby introduced, is about breast cancer and particularly diagnostic development in the RNA space. These are my disclosures. We have developed a patented predictor for sensitivity to endocrine therapy, and of course I'll be talking about that today. Okay, and this is the title of the presentation. All right, so just a little bit of background about breast cancer. Hormone receptor positive, HER2 negative breast cancer is far and away the most common form of breast cancer. It's almost 3/4 of invasive breast cancer are of that type. And endocrine treatment is really the backbone treatment for that type of disease. And in the adjuvant setting, which is sort of after initial treatment, it's used to be for 5 years, now it's recommended for 10 years. It is a long-term maintenance treatment, and should the disease relapse, it is pretty much the basis of combinations of treatments from then on. So it's very important to be able to predict the sensitivity to endocrine therapy and to be able to put it in the context of risk so that you can make decisions about, well, do we give chemotherapy? Do we combine the endocrine treatment with something a little more toxic but potentially more effective, et cetera, et cetera. So the other thing about the estrogen receptor pathway in breast cancer is it's far and away the most comprehensive influence on the transcriptional profile of breast cancer. I like to say there's just tidal surge, a storm surge of transcription, wave upon wave, that is related to estrogen receptor binding to DNA and activating transcription of other transcription factors, et cetera, et cetera. So one of our objectives in the development of a diagnostic approach was to take advantage of that knowledge and to try to understand, can we find the most reliable, robust, pre-analytically, analytically, and clinically relevant selection of transcripts that represent this biology, and in a way that doesn't consume a huge amount of analyte, so that we can really just get at like a thermometer. Basically, how hot is that pathway running? Can we get a basic measurement of that, and then we can make contextual inferences about clinical decisions with a measure of one of the fundamental biological effects. Okay, summarized all of that. I think you probably had plenty of time to read it. So when we were doing sort of proof of concept work with Affymetrix gene expression arrays back in the day, we published this work, more than a decade ago now, where we looked at pre-treatment biopsy samples, ran microarrays on them from patients with hormone receptor positive HER2 negative breast cancer who were receiving 6 months of chemotherapy, then surgery, and we'd developed this way to measure the residual cancer burden after the 6 months of chemotherapy, which is highly prognostic, how much disease is left. And it's a quantitative measure. And we're looking at the interaction, if you like, or relative effects of the response to chemotherapy. 0 means there's no cancer left, and 5 means there's a lot of cancer left, versus a prediction from the gene expression profile. This was based on 200 transcripts at the time off of the Assay data, where we looked to see was the average of those genes as a predictor of a measure of the activity of ER-related transcription. Was this in any way related to the risk of relapse over the next 5 years? And this is on a log hazard rate, where pink is a very high risk relatively of relapse within 5 years, and blue is a very low risk. And what we could see, obviously, if there's no cancer left, that's a very good sign for the future, and with hormonal therapy over the next 5 years, it was pretty much a low risk across this relationship. And with a lot of disease left, perhaps hormonal therapy is not going to be able to control that risk. So that may or may not be may or may not be manageable. But there was this blush of interaction between the two treatment effects, and potentially suggesting that we could identify a group of patients where we can start to understand the balance between what chemotherapy has done and what endocrine treatment is likely to do for that patient. And so essentially putting together the parameters of risk to build our understanding of how to develop a predictive assay. So this is the very simple, complex concept of what the sensitivity to endocrine therapy index is. We built off of this and then started to develop an approach towards a customized assay that might become more of a diagnostic approach. And so we know that both progesterone receptor and estrogen receptor are very closely related from a transcriptional point of view. PR adds a little bit of information, but actually in the relapsed metastatic setting, it adds a lot of information about the activity and the sensitivity to treatments. So we looked at the genes that were basically highly correlated with the expression of both estrogen receptor and progesterone receptor genes. And so I liken this to, we're measuring the water that's coming out of the faucet. And the tap for the faucet is the estrogen receptor binding to DNA and turning on this whole process. That's all we're really trying to do. And so the most important parameter for us in terms of this research exercise or development exercise is to reduce the amount of variance or noise in any of the measurements that we're making, and to combine them in a relatively simple way so that they are providing on that, so that when you say it's 70 degrees outside, it's probably 68 to 72, it's not 60 to 80. We want to have a fair degree of precision. So how do we get to a customized assay with just 18 transcripts to represent the transcriptional product of these two receptors? We put all our efforts really into a series of studies where we looked at all the pre-analytical and analytical variables that we could consider, did some nested transcriptional profiling of the samples from those experiments, and then basically ran through a pipeline to get the most technically reliable transcripts that we could from ours. So they had to be associated with the expression of both receptors, but they had to be tight under a multiple of pre-analytical and analytical conditions. And then the only other parameter here of relevance is that any gene transcript that was clearly related to proliferation based on its known biology, we eliminated from the list, because we know there's a known paradox between proliferation and risk and endocrine activity in a breast cancer. So these were the 18 transcripts that we selected, and these are the box plots of their expression values. And we also selected, using a similar approach, 10 reference genes to try to capture the approximate range of expression values from the Assay data of our target genes. So we really went perhaps a little bit overboard on the number of reference genes, but we really wanted to have a robust measure because of the average of multiple. And so in terms of developing this and looking now at going backwards at the data sets that we used to select the genes, we're now looking at the finished average of the target genes subtracting the average of the reference genes, all on a log scale. And we're looking at different parameters. So for example, this is the target minus reference genes for intra-assay replicates. That was every step in the Affymetrix gene expression pathway where we could make a split and do a replicate. This is intratumoral heterogeneity. This is 3 selected punch biopsies from around the tumor in 51 tumors. This is looking at inter-sample types. So this is, we take a cytologic sample and then a subjacent tissue piece from fresh breast cancers to try to understand these are differences in, these are the 2 most common types of samples that are obtained of a tumor in a clinical setting, particularly in metastatic disease. It could be one or the other. And they also have different cellular components. We're measuring what we think is in the cancer cells, but they have different components at the stromal level in cytology, scrapes, and tissue biopsies. So good concordance there. And then looking at 2 technical platforms, the U133A on the Affymetrix chip, and then their more updated higher density +2, minimal bias between the 2, but strong reproducibility across variations on the same theme. So we were comfortable moving forward with that, and then we were interested in trying to translate that assay into something that can be used in formalin-fixed paraffin-embedded tissue, which is the routine biopsy sample in the clinical pathology world. And also, we took the opportunity to take our premise, which is that this is measuring how hot is your endocrine pathway running in your cancer cells, right? A measure of activity, but also to then put that, essentially account for it, adjust for it based on your actual baseline risk, based on your baseline presentation of disease. T is the size of the tumor, N is the number of involved lymph nodes. This is all standard clinical pathologic information, very much involved in the planning of treatment for a patient. And then RNA4 is just measuring the 3 main receptors, ER, PR, and HER2, and a simple proliferation measure, aurora kinase. It could have been Ki67, but we chose aurora kinase based on analytical performance. And so this is your basic subtype of your cancer at a molecular level and your T and N burden of disease. So we want to measure how active is your endocrine pathway. That may be by itself important, but we also want to measure how active is your endocrine pathway in the context of your baseline risk, because that might provide another nuanced view of how to make a decision for that patient about what treatment to use. All right, so there's baseline prognostic index and our set ER-PR index, and so we combine those called it set 2-3, and it's just the weighted sum of those 2. And we shared the reference genes between the 2 indices. And in our MD Anderson experience with a large cohort of 10 years, this is the patients who have a moderate amount of residual cancer after 6 months of chemo, and this is the patients who have a lot of cancer after 6 months of chemo. So we've eliminated from this analysis the patients who did really well on chemo. As the risk relative to when there's basically minimal expression or activity of the set 2-3, we can see that as the set 2-3 rises in both groups, the risk of relapse drops. So it's adding information to the chemotherapy response, independent information. And similarly in that same cohort, if we look at the molecular subtypes, the genomic subtypes of hormone receptor positive HER2 negative disease in these same patients, luminal A are the most indolent, and luminal B are a bit more aggressive, and they have a different prognosis. They're very well studied. And we saw that taking these samples and splitting them to each of those categories, whichever one that patient's tumor fits with, we can see the same thing, that even with the molecular subtype known, this is still adding information as the set2-3 increases, the risk of relapse decreases in parallel in each of the subtypes of breast cancer. And we did in silico using adjuvant array data from one of the national trials, I-SPY 2. So this was an in silico analysis independently performed by the clinical trial group. We found the same thing, basically. Okay. Within our MD Anderson experience with the Affy array data, we were able to perform an analytical experiment where we looked to see of these two components, the baseline prognostic index and the SET-ER-PR index, that add together to form the set2-3. So those are the two components of set2-3, and this is the response to the chemotherapy, where 0 is excellent and 5 is the worst. In this multivariate model, what happens if we substitute one of the components? So in our baseline prognostic index, we had tumor-sized lymph node involvement and the RNA4. What if we substitute the RNA4 with one of the other signatures that's out there, well known, and actually all of these have a clinical diagnostic format that's used in practice. So what if we substitute PAM50 or recurrence score Oncotype DX or the EndoPredict? And this is in silico from Affy data. This is not their actual tests. And what we found was that it didn't matter. We could take the RNA4 and we could substitute it with any of these prognostic parameters. The model still works. The SET-ER-PR is still adding independent information, and the RCB response to chemotherapy is still adding independent information. Pick your favorite, right? But if we do the converse and we substitute instead the SET-ER-PR component, the endocrine transcriptional measure, we keep the RNA4 baseline prognostic indexes as intended, but we substitute the SET-ER-PR for PAM50 or 21 gene recurrence score, any of these, we see the model doesn't work quite so well. We basically just have prognosis, and this additional prognostic information of these replacements isn't really adding to the model. So just showing in another way that these two components are quite independent from each other. One of them could be readily substituted, but the other is somewhat unique. So with that in mind, we wanted to be able to switch to a platform that we can apply to FFPE, formalin fixed paraffin embedded routine biopsy samples. So we did a lot of comparison studies where we take a patient tumor and we finally mince it up and we randomly assign it to different conditions for a lot of these studies. And this paper from our group, published in 2020, summarizes a series of experiments looking at fresh versus FFPE tumor, looking at FFPE sections with purified RNA, or the simple homogenate method, which can be used with the xMAP QuantiGene workflow. So can we eliminate the need to extract the RNA, QC it, and instead just work straight off a homogenate directly off the unstained formalin fixed paraffin embedded section? Why would we care about that? Well, in pathology, that's a huge separate. Standard anatomic pathology isn't going to get into the field of QCing RNA or DNA. That's a molecular diagnostic test at that point. It goes down the road. Who knows how long it'll take to get there? There's a whole logistical complexity apart from that. Whereas pathology does immunohistochemistry, it does an in situ hybridization. This is just a variation of in situ hybridization if you don't have to QC the RNA. So that was an important study for us to do for practical purposes. Cores is the biopsy you might get. Resection is the tissue removed at surgery. You need to know that you're going to be able to translate between those two because you don't know how the patient's going to be managed. Your test has to be able to work on either. Long-term storage of long-term FFPE versus RNA from the same sample that have been stored at the -80 for a very long time. That's not relevant to the patient you're seeing next week, but it's very relevant to the clinical trial samples from 10 years ago that are going to build the evidence that make the case that your test is worth anything. in terms of clinical meaning. And then same thing for frozen tissue versus a corresponding FFPE from the same patient's tumor. And then some precision assays looking at two persons each alternating over 20 weeks, analyzing a section each over multiple tumors to see how stable our results are. And then finally, what is essentially a limiting dilution assay to get a sense of sensitivity and also to understand quality control requirements for reporting the result. So, looking at FFPE samples and looking at the QuantiGene from the FFPE with the frozen result from the same patient's tumor with the microarray, which at the time was our gold standard. We saw in 40 cases, and we did exactly the same approach in the same cases with the nanostring platform, and we saw both of them very, very similar result. Good concordance, a simple linear regression approach could perhaps calibrate between the platforms. We did that, and then we tested on 23 additional other tumors, and we found that the calibration held. Looking at the reproducibility in terms of with repeating the assay basically with a frozen sample, we saw that there very strong concordance on the microarrays and on the QuantiGene using a corresponding FFPE sample and on the nanostring. So basically, in a race between nanostring and QuantiGene using the xMAP, we see no difference. We made the choice for xMAP based on some preferences and some workflow logistics that we thought would be advantageous, but this is absolutely equivalent to the nanostring for FFPE gene expression measurements. What about the purified RNA versus the homogenate directly applied to the unstained section? We saw outstanding concordance, and this was another group of tumors where we did this, and we've consistently seen this. So this motivated our decision to be able to just keep this within just general pathology. Let's just simplify the workflow. And even with old blocks that were sort of 8 to 10 years old from the archive, we saw the same thing. So a very stable platform for us for our intended use. Finally, showing here, if you make the decision that you're not going to purify RNA, that means you make the decision that you're not going to know how much RNA you're loading from the patient sample. So how are you going to QC that? How are you going to know when you can or cannot report the result? And our limiting dilution using FFPE and then just serially diluting 1-to-1, basically starting at 125 nanograms loading from FFPE RNA and going all the way down to about 3 nanograms. Sort of an absurd project to do. But what we use is we use the average of the reference genes, of our 10 reference genes. And we look to see, we've got the frozen sample that was run on the array, and we've got the corresponding FFPE that's been serially diluted, and so each measurement from each sample is moving to the left, right, because there's left starting material. At what point do the results really deviate from what they should be compared to the frozen standard? We did this in a series of tumors, and we've color-coded them for each of this is the multi-gene signature, the individual genes. We've color-coded them according to their, in the frozen sample, their value. So the higher expression ones are darker, and the very low expression ones are lighter. It's a visual, just helpful. And so we're seeing the problems with deviation in the low expression range of whatever we're measuring. But we found fairly consistently that if the mean of the reference genes was below about 3, we conservatively said 3.5 on the log scale, then we could safely assume that the result was correct. So we took an empirical approach to our QC threshold, and that's what we've been using going forward. And this just summarizes all of the various experiments from that paper for that FFP transition over to QuantiGene. And so we made a table in the paper to summarize all the results if anyone's interested. The next study was an inter-laboratory study where each laboratory, it's just basically a simple split of FFPE sections with the corresponding H&E sent to 3 labs. One was us, and then B and C were new labs, clinical labs in other parts of the country. And we got a lot of really great support from Thermo Fisher, who placed units into these labs, did the proper training of each site, and B and C were doing it for the first time. Everybody did the 60 tumors, and then they got the same 60 tumors again and did them on another day, and we did batch controls of the various reagents. We sent out the reagents, so we made sure there was multiple batches being used of everything. So we're able to look at intra-lab reproducibility of measurements of these 60 tumors on 2 separate occasions in each of the labs, and we found very, very strong concordance in each of the labs. And again, I'll point out B and C really had no experience. They got the training, we gave them 3 practice samples, everybody seemed to be about right, we got on with it and got the study done. So easy adoption. And then for the inter-laboratory reproducibility, this is showing the average of the 2 measurements from each lab compared to the average of the 2 measurements from the other lab, so each combination, A versus B, B versus C, A versus C, the story's the same. Excellent concordance of this assay on FFPE in different labs and between different labs. So then this gives us the confidence to really look at the clinical validity. And so we started out with a registry study. We've already shown some retrospective work, but we started out with a registry study where we collected samples. This was an ongoing study. We were doing Affymetrix U133A arrays. We went ahead and just did the QuantiGene on the residual RNA from those samples, and they were from just under 300 patients with hormone receptor positive HER2 negative breast cancer. And what we found, we then just tracked them. Doctors did whatever tests they wanted to do. They treated the patients however was appropriate, and we have about 5-year follow-up, and we can see in the overall 300 patients, the set high using our predefined cut point and low were well separated, and most of the patients got chemotherapy, and of the patients who got endocrine therapy, very few of them were set high. But basically we're seeing everybody got endocrine treatment, and we're seeing a nice separation in prognosis between those 2. In our retrospective study, we had calibrated this to adjuvant arrays with the I-SPY work. So we used that same calibration formula for the adjuvant array data from the ACOSOG Z1031 trial, which was an interesting study because patients got endocrine treatment for 4 months, and then they got surgery. So they didn't get the chemo first. They were allowed to get chemo later, but they started out as an intent to treat with endocrine therapy alone. They had stage 2 to 3 disease, which is more extensive. And what we found was that the pharmacodynamic suppression of proliferation on a biopsy taken at 2 to 4 weeks. There are 2 measures here. One was the 2 to 4 week measurement, Ki67 less than 10% of the tumor cells, and the other was what has been adopted as complete cell cycle arrest, which is a Ki67 less than 2.7%. And we saw a significant increase in those pharmacodynamic responses in the patients whose baseline sample had a high SET2/3 index. We did not see a prediction of the response pathologically at the 4-month period, but we did see a strong prognostic difference in favor of those that had the high SET2/3 in terms of 8-year survival. And you'll notice endocrine treatment's given for 5 years. At that time, they're separating nicely for 5 years, maybe for a year or 2 after, and then the story's over because the treatment finished. So predicting an early pharmacodynamic response and predicting long-term survival benefit for those that have high activity of the endocrine pathway measured by this assay. And so then finally, we have presented a few months ago at the San Antonio Breast Cancer Symposium, this is a viewable online poster, we looked at in silico at our assay in the SWOG S8814 trial, which had performed first of all, it had performed and published the Oncotype DX, which is the standard, most favored test in the marketplace. But this was node-positive disease. We don't normally do that, any of these tests. And these patients with node-positive disease had been randomized to receive chemotherapy, followed by endocrine treatment for 5 years, versus just endocrine treatment. So no chemotherapy at all. So first of all, because we're dealing with an RNA-seq FFPE data set, we did a formal calibration before we started the project. This is the set-to-3 measured with the QuantiGene in 106 FFPE samples, and this is the RNA-seq performed on those same RNA 106 samples. We see a very nice calibration. This is a ZER-PR prior to any linear regression calibration. You can see it's already got a very nice, very little scatter, but there's a little bit of bias and there's a little bit of gradient shift. So the linear regression cleans that up nicely. And even when we account for the BPI as part of the index, we were very happy with this concordance. So QuantiGene and RNA-seq from FFPE, very concordant. We've done it on 2 types of microarrays and RNA-seq, so we're very happy with that. And these were the results from the patients, about 283 or so samples from patients on the trial, and the high set using our predefined cut point was very favorable prognosis, particularly over those first 6 or 7 years. So 5-year treatment. The other interesting thing we found from this was that compared to the Oncotype DX, there was really no correlation between the two. The cut point in node-positive disease for the Oncotype DX recurrence score is 25, and we found that half of those of 25 or below were high set, and half of those that were above 25 were high set. So really really quite surprisingly different and potentially interesting. So when we look at these 4 combinations, we found that when both of the tests had a favorable outcome, there were very few events over the first 5 years during the course of treatment. And even in the patients, the half of the patients who didn't get any chemo at all, just tamoxifen. So that's quite encouraging. Obviously, if they were both unfavorable, this was bad news, prognostically, in overall and in the endocrine treated. And if they disagreed, it didn't matter which way they disagreed. The outcome was the same. That's interesting because it suggests that maybe for those patients, there could be different approaches. Because one is imagine you're sort of poor risk, but highly endocrine sensitive. Well, targeting the endocrine treatment with additional treatments would be a very smart thing to do. And then the converse, one could comply this. And this is really one of the fascinating things, because in this field of breast cancer, most of these signatures, they're just very highly correlated. People say there are variations on the same theme, but this assay is actually different. And then I will say that we're not able to show it, but we have some data that's going to be presented at ASCO meeting in June that's embargoed until the meeting, so we couldn't show it. Where we've done this now in a 600-patient study from 25 years ago, using residual RNA from those tumors and performing the assay on the MAGPICs, the T200 using the QuantiGene method, and we're very excited by those results, but we can't talk about them yet. I say, to conclude, a selection of analytically reliable transcripts from a research genomic platform and their combination into an algebraic formula leads to a custom assay that is analytically reliable on this platform. We're very happy with the translation to FFPE. We see strong inter and intra-laboratory reproducibility. We've been able to accurately calibrate between the QuantiGene on the xMAP platform, between 2 types of arrays, as well as RNA-seq in fresh samples and in FFPE samples. This has greatly accelerated the clinical development of our attempts to develop a diagnostic, because it really opens the doors for all sorts of data in silico analyses from clinical trials. And these days, a lot of the clinical trials are only doing RNA-seq because it's a bit of everything. So this really opens up for the ongoing and future clinical trials, it opens up everything. And then testing of archival samples from randomized clinical trials is already underway, and some of the first results will be reported in June. Regarding the SET23 assay for breast cancer, technically robust in FFPE tissues, independently prognostic compared to other contemporary prognostic signatures, and adds independently and significantly to leading contemporary prognostic and predictive tests. And so that's potentially a clinical utility approach. And we've just recently established this in the CLIA laboratory to support prospective clinical trials and in commercial development. And our commercial partner has established this in the CLIA environment and is providing testing results to currently one prospective clinical trial and is looking to expand that effort. And I will leave you with a list of thank yous that is probably longer than an Oscar acceptance speech. But I do want to point out that most of our research lab members who really know this assay inside out and were very involved in the development of this are in the room and would love to talk to anyone who's interested. I also particularly want to highlight Stefan, Colin, and Tiffany from Thermo Fisher, who have been really amazing supporters to us from the very beginning of this exercise. So thank you very much.
Thank you, Dr. Simmons. Any questions for Dr. Simmons? Actually, I have one question. I remember that in a couple slides ago, you show the comparison of the disease-free survival on a 5-year stage of all patients versus patients treated with ET only. If I understand this correctly, the tamoxifen only, am I not seeing it's similar? Am I understanding that? Is it because there's a resistance? In other words, did you look at the BCL to Bax ratio with these patients to see why, if there is a true resistance here of tamoxifen?
We haven't. We don't, in this setting, we're coming to the data owners for an independent blinded validation, so we're hands off in terms of the data.
Right.
They're going to look to try to understand, what this means. One of the paradoxes of the endocrine activity, and one of the reasons why this chemotherapy interface is quite confusing to many in the field, is because the endocrine activity, many of your BCL2, many of your survival-related, somewhat anti-proliferative, and even things like MAP-tau that obstructs the binding site of taxane chemotherapies. These are part of the tidal surge of endocrine activity, and so it is. That's why we've really tried to take a modular approach, knowing that in our stage 2 to 3 setting, until very recently, it was chemo then endocrine.
Right.
So if you're giving both, how are you going to break down the puzzle into different pieces? And that's really the motivation for us at the beginning to say, we're going to separate in the signature development phase. We're going to take endocrine activity that is not related to proliferation, because we know that tells us a lot about chemotherapy response.
Right. Yeah.
And we want to basically be able to deconstruct this into modules and then put them back together into a predictive algorithm. So I think you've got at the heart of what we're trying to do, without getting into the details of how resistance and why it might happen in a patient. We just tried to do it across the population conceptually from the beginning.
And I apologize, maybe I missed it. What stages were these patients? Were they more early stage or late stage?
Later stage. So they were postmenopausal patients who got chemotherapy, who had surgery first, and there were lymph nodes involved.
Yeah, great talk. Thanks very much. So your panel was, what, 21 markers?
31.
31,
Yeah.
okay.
Yeah.
Because I'm kind of looking at this from more of a bioinformatic perspective, because we have other users that are also looking at other diseases that have multiple markers, and they're trying to weigh, how the shifts in those markers can help predict the occurrence or resistance to a non-infectious disease.
Yep.
And I'm trying to figure out how you're handling that 30-something marker data to get a score. Because for you, it's with or without hormonal treatment or something like that. So for these other applications, it's more like, are they going to be more susceptible or not to going down a certain disease path? And I'm trying to grasp how you're, getting those scores might help for some of these other types of analysis.
Absolutely. So for this indication, we took the approach that we're measuring a pathway that influences transcription. So what we want to do is get the noise out of the system. And so we just evaporated many of the we had right down, right? And we actually did something similar for a PI3 kinase related transcriptional signature with colleagues, friends who've published this, even patented it, 300 gene signature. We applied the same pipeline, got it down to 9. Because in both of those settings, we're saying, look, we just want to measure how active it is. But for the example, let's say trying to develop a chemotherapy predictor, now we're in the empirical model, responders versus not, very much like what you're describing it. Yeah, in your example. And in that setting, you're still talking about signal to noise ratio, and sometimes the signals are quite low. And so our approach has been to say, well, let's get the noise out of it as much as we can. So if there's a low signal, at least we can see it. And so that's very much work in progress, but we've taken that approach on the chemotherapy prediction side to try to see, can we get a way to reduce down from what would otherwise be like a random forest model with hundreds or thousands of potential signals, all of which are making noisy mess in the background. And when you translate those to somebody's biopsy, fixed in formalin, bedded in wax, it's all going to go away, And so that's the approach we've been having. It's challenging. With that setting, we've tried to get a quantitative model. So we don't get serial measurements, but we do get that chemotherapy response, and we have it on a continuous scale with this residual cancer burden index. So we've looked for things like correlation and inverse correlation. We're trying to make instead of relying on arbitrary cut points, we're trying to let the continuous nature of the outcome and the data give us a bit more of a measurement approach. But I'll re-emphasize, I think that's still obviously work in progress, but I think that the confidence that the custom platform that we plan to measure, if we do our job right, will not have that noise that we've been trying to avoid all along, and will be very, very linearly relevant to the discovery platforms in terms of calibration. Gives us a lot of confidence. That's our job, is to find the right set, and it's really hard. Yeah. Great question.
Any other questions for Dr. Simmons? Yes.
Thank you. It was very interesting. I just have a question based on your experience and your expertise. We saw a good correlation between the Affymetrix gene expression data versus this Plex application of the Luminex assays. If you would put them side by side, what would be the pros and the cons of each technology according to what you have seen?
Yeah, you could ask that question related to RNA sequencing as well, and it would be equally relevant. It comes down to philosophy, and I think as a practicing pathologist and knowing to some extent the ins and outs of diagnostic testing and diagnostic QC, regulatory, all of that stuff, although I came into this field much younger with the mentality that we'll do everything and it'll all just come out of the data. I think that the economics, the timeline, the complexity, and the regulatory aspects are, although some believe we'll just sequence everything from everybody and we'll work it all out, I think if you can be there the day after tomorrow with an actionable result that's reliable and it's going to be regulatory, that's important too. And there's two ways of looking at it, and there are two types of uses, and I think we need both. So, did that answer your question?
I was just curious to know, because we see both options, and both seem to work well. But I just wanted to get your thoughts about what is good about this, what is not so good about this.
Right. I think what's really good about this is if you know what you want to measure, and you know you've done the diligence on a more comprehensive platform. Maybe it's a bigger profile on a similar platform, or maybe it's global arrays or RNA-seq. So long as you have really vetted what you're measuring, then you make the conversion and say, look, we need something that's going to be able to work everywhere, and it's going to be economic, it's going to be timely, it's going to be efficient, it's not going to cause QCs, and your average clinical technician is going to be able to learn it like this and do it, and maybe it's even open to some further automation for some of the benchtop steps. There you're going to have other choices. And I think one of the key points I wanted to make in this experience, although this has not really been widely published in the RNA space, this works. And it works as well as one of the leading technologies in terms of adopter use. We've been nothing but impressed. Sure.
Any other questions for Dr. Simmons? Okay. Thank you, Dr. Simmons. Really appreciate your talk.